Good evening, everyone. Thank you for joining in this webinar, which is only for an hour, followed by 15 minutes of Q&A. So the time is tight. And it's a really interesting topic on the exploring the different options in enteral nutrition. The other two speakers are the real experts and with more experience than I have. So I've decided for my introduction to just give a brief general introduction to what I think is the way we should be looking at nutrition and the gut health. And then I'll just follow a little bit of my second part of my presentation with some thoughts on pediatric bone marrow transplantation. So as I said in my bio, the overarching hypothesis that I think about every day is that there are three major players which interact very intimately. And the first player is the mucosal immune system, which is determined by our genetics. And it's always nice to remind ourselves that 70% of our immune system actually resides in the gut. And that's not surprising because the resident microbiome is what it interacts with. And these two guys are really, really, like I said, intimately linked from day one of birth and throughout life. Because our GI tract is quite long and has distinct ecological niches, which adds more challenges because a lot of different functions are happening in different parts. And the third player, I think that we have to, of course, always keep in mind is external triggers. And for me, I put nutrition under non-noxious because without nutrition, nothing else, there'll be no life. But at the same time, there are noxious triggers as well, which are infections and allergens. And it's a combination of all these different players that actually determines gut health versus disease. And so you can't ignore one versus the other. And I think this is becoming much more clearer since the microbiome has taken central stage in the last 10 years of research. Now, I was thinking in terms of therapeutic manipulation in a condition or of any clinical condition, you really can't do much with the mucosal immune system because you can't change your genetics. And you don't want infections and allergens there. So the two things that are amenable to manipulation is the microbiome that we're learning more and nutrition. And I just feel that in previous traditionally, we have thought about nutrition and its impact on human health, which is absolutely correct. But I just feel as we move forward that nutrition, it's the same nutrition that's also feeding our microbes. So I think in terms of identifying human health, the benefits of nutrition, we now cannot ignore what the nutrition that we have on a daily basis, what it's doing to our microbiome as well, because the microbiome directly and indirectly affects health. So this is another, I think, dimension, another prong of nutrition that I think is, again, becoming much more important as we go along. I just thought, again, from a general background, in terms of man's evolution on this planet, how things have changed. And I just wanted to go back just to post-industrialization, what we humans have been doing. We have done some lots of good things. Presence of clean water, not everywhere, but still our dietary habits have changed. Some things were quite, absolutely, changed the landscape of population health, like pasteurization, sterilization. Now, much more in the last 50 years, we're getting cold storage deliveries, long shelf life of our food. We all know about processed food and colors and emulsifiers, are being added. Lifestyle has changed as well, largely sedentary. Medicines are also a big invention of the last 100, 150 years, vaccinations, and of course, antibiotics. And all, so I just feel that the effect post-industrialization we are having on our microbiome and on human health has actually been much bigger than maybe in the previous years. The advantages of all this, of course, have been there's some very good news. We have less infectious disease compared to, unlike COVID, of course, they do emerge new infectious diseases. But in the last 200 years, that's made a big impact on populations. Infant mortality, being at the Institute of Child Health, is a big thing that everyone is proud of. And we've increased man's lifespan. Again, this is all very good. But, the outcomes of how we go forward are still unclear. How all these changes are impacting on our life course. Childhood obesity, for example, as one example. The general population, of course, old age. Then that, again, the nutritional demands in old age are, of course, again, something that we really need to concentrate on. As our immune system declines, the bacterials will decline in their own form. And then, we need, that's another focus we need to really spend a lot of time on. One last comment about these general changes is urbanization. This is a great paper where if you look on the right-hand side, this is done in Brazil, and they went looked at different populations at the same altitude and the same temperature, going from a jungle environment all the way to city and middle class. And then if I can direct you to the left-hand side, and of course, the more urbanization you get, the more industrial materials, paints, for example, and that we use in construction, whereas the natural building materials are changing as well. And we're not only impacting on our own health, but the pets and the planet Earth itself. So big changes, and a lot of these things we just still do not understand. Coming back to diet and the microbiome, this is, I think, one of the first important studies that I remember, which I think made an impact, was a study from Paolo Lunetti in 2010, I think, published in PNAS. And they looked at the microbiome of children in rural Africa, which is in A, and the Western diet in B. And you can immediately see there that the landscapes are very, very different. And of course, it all comes down to, a lot of it is, of course, nutrition. And what's quite clear is if you look at the top greens, Prevotella and Zylobacter, is because it's more plant-based diet, and also the, plantains, for example. So yes, our diets have a major impact on the landscape of our microbiome. And I think that's where, and this is without actually looking at the health of the individuals. So this is the first collision of how microbes are actually so critical for helping us digest food that we actually cannot do ourselves. And we know an awful lot about that now, but not enough time to talk about it today. I just wanted to raise one comment here, which is differential resilience. And what's happened since 2010 is that one thing I think everybody in the field will know is the bacterial byproducts called short-chain fatty acids, which are meant to be anti-inflammatory, very good for good general gut health. What people are finding is that even though we might have the right microbes to make these good short-chain fatty acids, but the number of species that do it is much less in the Western diet compared to, let's say, Africa. And the consequence of that is that when we get perturbations in any clinical condition, there's more propensity to collapse. Because if you only have 6 species that make butyrate, for example, and they go missing, then you're in real trouble. Whereas here, I think there might be a lot more. So it's not just looking at the microbes and their byproducts, but this resilience is against something which I think we learn a lot more as we go into this nutritional microbiome axis. One word of caution as we move forward, that although there's a lot of microbiome data out there, got to say that majority of it is coming is overrepresented by the States and Europe and China. And just as an example of this overrepresentation from America, that 40% of microbiome studies have come from America, but they only represent 4% of the world's population. So again, some of the therapeutic manipulations that we might have in future in mind, that there's still at least 120 countries that there's no information from. So a lot of work to be done in this field. And I just want to say this is a reminder. This field of nutrition microbiome, I think, is in its infancy, and hopefully I look forward to seeing new information becoming increasingly available in the coming years. Since I'm talking about the microbiome, I just wanted one last slide to show you that to visualizing this microbiota. And I think it's a fun slide to share. The first time it was ever seen was in 2008. So it's actually, again, very recent, a paper by a Swedish group in PNAS. And it is a mouse model. And here in blue, you can see, I hope you can see in figure C with my cursor. This is the colonic crypts, and the pink staining is for the bacteria. And what you see very nicely in a healthy gut is this black staining or lack of any color, and that's real demarcation where the microbes are not coming close to the gut, and that's because mucus is there and it's actually keeping them at bay. Yes, but products do diffuse and have an impact on the gut health. But the minute the investigators actually mutated the mucin in the top panel, you can see they get crypt hyperplasia, and all the microbes have just moved and have settled on top. And I think this was just visually really nice to see that these guys do exist, not just in a fecal sample by 16S. you can actually view them, and then what can happen in disease. And the minute they come on top, they start entering into the gut lining and bacterial translocation happens. So this is 2008. This is to 2021. Pictures like these on the left have turned into single cell transcriptomics. And now, it's just amazing the amount of information that's coming out. It turns out there are 4 types of goblet cells in the gut, but there's only one type that makes this mucin. So amazing progress. And I think, again, it will really change how we think about the crosstalk between microbes and the gut itself. Okay, so the work I want to really spend in the last half of my presentation is the impact of the gut microbiota on clinical outcome in transplantation. As I know, Niruz, the second speaker, is going to go into some detail in adult stem cell transplantation. I'll keep it very simple. My main point on this slide, which I have borrowed from a Nature Review article, is that when somebody comes in, a child with leukemia, and you have to eradicate the patient's immune system, there are all kinds of different manipulations, including different extensive irradiation, chemotherapy, radiotherapy, antibodies. So to deplete the immune system, and before you can put the donor's new stem cells. And once you deplete the immune system, of course, the microbes in the gut also have nothing to defend the body against those microbes. So that's where antimicrobials come in. So these patients are on very, very heavy antimicrobial usage because they are vulnerable. So just quickly to show some data from our recent work, which got published in June this year. So coming back to Great Ormond Street, most of the patients are treated for leukemia, immunodeficiencies, childhood, or metabolic diseases undergo transplantation. There are about 100 transplants a year. 70% are allogeneic, so it's an unrelated donor, which means 30% is where they might be getting CAR T cell therapy or gene therapy, where their own immune cells are being manipulated ex vivo and then being cured. So it's an exciting time. There is obviously greater increase of autologous transplantation as well. And in this paper, the first author, Amber, it was her PhD work, investigated 64 patients, and the age range from 6 months to 14 years. And on the right-hand side, I've got a slide just showing, here it says day 0 of at transplant. So you can see just a few days before, a week before, and 2-3 weeks after transplant, that how many antimicrobials, they go up to 15 administrations in an individual, how much you have to give to the patient to keep them because they don't have an immune system. But we can also understand the detrimental effect on the gut as well. What I think this is an interesting slide to share with you. What Amber found was that we could actually, point number 1, I think is really important, is when patients come at admission, their microbiome already is not as diverse as that of healthy children. So there's a window of opportunity there, which I will hopefully come back to at the end of my presentation. But when they arrive and the sample is checked, what Amber has been able to do is to show there are 3 different community types. The blue here are the healthiest there are, but not as healthy as healthy children. The orange is the second community type, which is less healthy, and then the gray are the least healthy. And if I move to the right-hand side, what happens in the first 5 weeks of post-transplantation, the microbiome collapses. So the healthiers become less healthy within a week. And what you find is that the patients, as time goes on, the worst scenario, community type, the gray, then starts getting more and more. So the longer the patients stay, this is 300 days in a hospital, in the clinic, that the microbiome never actually recovers. So this is something, again, we really need to address. You can't have patients in there for so long. And what was interesting is these 3 different clusters. The worst one we found associated with a high risk of viremia. But what's important for today's talk is that it was also the use of TPN. These patients did worse, whereas the ones that had a good microbiome and stayed in the top state being on enteral nutrition. So I think that is something really, really important to highlight today. So I would like to say that the current challenges in transplantation are the post-transplantation with no immune system, at least for 2-3 weeks, there are no neutrophils. So the neutropenic patients are very vulnerable to infections, especially if you've got microbiome there, which means increased translocation, sepsis, and death. And death still remains at a 10 to 15% of patients. And that, again, is something we all have to work towards and to try to reduce. For a clinician, I guess, to mitigate this, absolutely, they have to use extensive antimicrobial usage to save their patient. But the consequence of that, of course, is that you're destroying the gut microbiome. And that means that the immune system is not being educated, or as it gets in graft, so there's slow reconstitution, which means prolonged hospital stay. So it's a bit of a catch-22. And I guess I don't need to provide evidence. If there's no microbes, the damaged gut because of chemoradiotherapy is suboptimal digestion. But our work and others actually have also shown that enteral nutrition is superior to TPN. So there is hope there. But how we take that forward is, I think, one of the things that I would like to discuss more today with you. So the question I have is, how do we modulate enteral nutrition in this group of patients to improve their microbiome, knowing that you can't give the microbiome when they're neutropenic? So this timing could be interesting, but also improve the health simultaneously. There are lots of things going on, clinical trials, people are using different antibiotics, they're trying to give probiotics, they're trying to give some substrates like oligosaccharides, potato-based starch, so I don't have time, FMT. But there is space in this area that we really can help our patients. And nutrition really has come up to be top. Just to finish off in the last couple of minutes, this is a paper that came out in 2022 this year. They've done it in mice, but they used a combination of plant and animal protein supplementation. And for me, what was interesting was that they suggest in this paper that animal protein on its own is is absorbed very quickly into the blood, whereas the soya-derived peptides take much longer, and also they have a real impact on the oxidative stress. And with this dual supplementation, on the right-hand side, they've looked at a lot of immune markers, and they see much faster immune reconstitution, for example, on day 21. With the double delivery, they see a much more NK cells, for example, compared to just the animal supplementation. So there is a lot of potential to discuss further. For my last couple of slides, the same group in last year published, they took post-transplant patients for 60 days, gave them a protein supplementation, and then at the end looked at those people who showed increase in muscle strength, mass, and those who did not. And the end message was, then they looked at their microbiome. So those that showed improvement in their on their supplementation in protein, actually, in your muscles strength and mass, they showed increased diversity of bacteria, whereas the other group did not. So it's really linking microbiome with nutrition, microbiome, and then the patient health. And this increased diversity also showed an increase in butyrate producers here. Just one quick comment that not every part of nutrition is good. Lactose actually drives enterococcus expansion, which is our antimicrobial resistant microbes, and this can be really, really not very good at all. And so lactose is a common nutrient, as this came out in Science in 2019. So I just wanted to highlight the positive and the negative aspects and how much more research needs to go onto it. So the emerging paradigm is that nutrition, forget about host health, directly impacts on the quality of your gut microbiome. And this in its own right is a crucial determinant of clinical outcome in transplantation. So I've changed the title that I started with, is I said how to best feed a hungry gut. I've added how to feed hungry microbes as well, and pre and post transplantation. So as I said earlier, the patients come in and their microbiome status is already not as impressive as healthy children. So maybe we should be promoting nutritional support pre-transplantation on diagnosis of leukemia and then by the time they come for their transplantation. Post-transplantation, I think it'd be quite nice. Should we do a combination of PN, which goes directly into the bloodstream nutrition, which helps the host, whereas should we do EN, which might just be more directed towards the microbes? For example, we don't want to give lactose in ENs because we don't want enterococcus to grow up. Should we also introduce at the same time some microbial products like short-chain fatty acids to help the gut heal in that neutropenic time when we can't give microbes themselves? Do they need some cytokine help, for example? So as we learn more about these different community types I mentioned, I wonder as we go along, the more we learn about the microbiome, could the EN and enteral nutrition also become more personalized? So a lot of work to do in this field, but I hope we are moving the right direction. So my last slide says here is from a paper actually from 2016, but I thought I'll share this with you. It says perhaps a more thoughtful modulation of the gut microbiota through alteration in nutrition before and after transplant may decrease the risk of development and severity of rejection, with little risk to the patient. And I think this is where we really should be concentrating. So my last comment for my presentation is nutrition, microbiome are so important that now we really need to bring scientists involved in these two areas of research to the same table to actually improve patient